Professor Maria Helena and Professor Gisele, because this is either a beginning of a new research line, because at present, this is about the characterization of bradykinin-potentiating peptides, BPPs, from Bothrops moojeni. So I call this lecture Old Molecules, New Targets. So although this has been shown before, we know that the formation of angiotensin II is catalyzed by the angiotensin-converting enzyme ACE. So as we can see, angiotensin II is a potent vasoconstrictor, and its formation is catalyzed by ACE, which is a carboxypeptidase that works on angiotensin I, which is by itself produced by the hydrolysis of angiotensinogen by the enzyme renin. So ACE is a hydrolase. And ACE, what ACE does is to hydrolyze the two C-terminal residues of angiotensin I and then produce angiotensin II, which is itself a potent vasoconstrictor. Although the characterization of ACE was really not straightforward at first, but it was known that it was a carboxypeptidase, so they resorted to a known inhibitor, which is L-benzylsuccinic acid, so this was known previously, and this served as a lead to guide the design of novel ACE inhibitors. So ACE does not only produce angiotensin II, which is a vasoconstrictory peptide, but ACE also degrades bradykinin. Bradykinin is a nonapeptide that is produced from kininogen by the action of the enzyme kallikrein. And bradykinin is hydrolyzed by ACE to produce bradykinin 1,7. So bradykinin interacts with the B1 and B2 receptors, which are GPCRs, and bradykinin is itself a vasodilatory peptide. So the hydrolysis of bradykinin to produce bradykinin 1,7 reduces the activity of bradykinin. So these peptides are called bradykinin-potentiating peptides, BPPs. They can be obtained from many sources. There are many animal venoms in which you can find bradykinin-potentiating peptides. For example, Brachycephalus ephipium contains a peptide called BPP-brachy-NH2, which is carboxyamidated, which is not very common among BPPs, but then it is there, and it does work as a BPP. Also, scorpion venoms contain bradykinin-potentiating peptides. And for sure, we have to mention Bothrops jararaca as a source of bradykinin-potentiating peptides. So these were known at first and characterized by Bothrops jararaca, by analyzing Bothrops jararaca. So when you first start looking at these sequences, then you start realizing that there have some conserved positions in the primary structures of these molecules. And this was actually published by some colleagues from Butantan. This is the DOI that leads to this very good paper where they analyze the structural conservation of some parts of BPPs. For example, you have a very well conserved pyroglutamate at the N-terminal. So this is quite common in BPPs derived from snake venoms. Also, you have the conservation of the C-terminal, which always ends with a proline, and usually a proline that ends with a carboxy terminus. So no modifications. Carboxyamidations are quite common in animal venoms, and these have no modifications. But also you have internal segments that are quite common, like PRP. This is a motif, or a tripeptide motif that is quite common. And also you might find tryptophan as the second or the third amino acid residue from the N-terminus. So when you analyze this primary structure of these BPPs derived from animal venoms, you see that they extend, or the sequence conservation extends way beyond the C-terminus. So you can find several conserved parts. And this for us, biologists, I'm a biologist myself, although I work at the Institute of Chemistry, I always like to say that I am a biologist and I think like a biologist. And when you do see sequence conservation, you always think that this has some relation to the activity of these molecules. Conservation is not there as at random. So conservation is there mostly because this might have something to do with the biological activity of these molecules. But then again, when ACEs when bradykinin-potentiating peptides were simplified to produce ACE inhibitors, succinylproline was used as one of the molecules for simplification. This was based on T-protide, which was characterized from Bothrops jararaca. And as we can see, succinylproline does contain the C-terminal proline, which is quite conserved among BPPs, snake BPPs, but it also contains some other parts, like, for example, this carboxylate part that interacts with the conserved zinc residue in the ACE active site. So this carboxylate does some ionic bonding with an arginine here, so this is quite important. There's a hydrogen bond between the oxygen in this carbonyl moiety, and also there are some pockets that were not being used in the development of these molecules. But then again, as we can see, this is based mostly in the C-terminal of T-protide. So besides the idea that these molecules are quite well conserved, just the C-terminal has been used for the development of these inhibitors. So this is all the strategy that has led to enalapril and the formation of enalaprillate, which has a very high affinity for ACE. It's like 1.2 nanomolar, the IC50, and based on succinylproline. So it's a strategy where you have several extensions and explorations of pockets that are quite interesting. So this is the optimization strategy that led to enalapril. Teprotade was reduced to its proline C-terminals and its interaction with ACE. So BPPs in snakes are thoroughly conserved, but then only the C-terminal is relevant to its biological activity. It doesn't make much sense to me. So that's one of the things that I think is interesting in this area of research. And then in 2009, a publication started showing that BPPs might have other targets besides ACE. One of the targets that was characterized was the enzyme arginine succinate synthase. So this cycle is called the citrulline-NO, nitrogen oxide cycle, and this cycle, it works or it operates in parallel with the urea cycle. So arginine succinate synthase, it has as a substrate citrulline, ATP, and aspartate, and then it catalyzes the formation of argininosuccinate, which then by the action of a lyase produces arginine. And then arginine can lead to the formation of urea, or also arginine can lead to the formation of nitrogen oxide. So this is vasodilatory. So it has been characterized that arginine succinate synthase is also a target for bradykinin-potentiating peptides, which leads to the idea that these molecules are have much more targets than simply ACE. In this paper, they have characterized the effect that this peptide, which they called BJ-BPP10C, has in the Km of this enzyme. So they showed that the affinity of the enzyme for the substrate citrulline and ATP are very are affected by the peptide at the concentration of 0.5 micromolar. So BJ-BPP10C affects the Km of arginine succinate synthase for its substrates. And then they demonstrate in the same paper that even at small concentrations of BJ-BPP10C, for example, 1 micromolar or 0.5 micromolar, the intracellular concentration of the amino acid L-arginine is highly increased in relation to the control sample, which demonstrates that indeed it seems that BJ-BPP10C is activating argininosuccinate synthase. And then it is leading to the production of nitrogen oxide, which they show in this paper again. They also use some inhibitors called MDLA, which is an inhibitor of argininosuccinate synthase, to show that after inhibition, this doesn't happen. So the peptide is apparently modulating the activity of ASS. This has been quite interesting, and this is what led to my interest in these peptides. So what is intriguing in this line of research, at least for myself, and as I told you, this is the beginning of an exploration, at least for me, is that it seems that there's more to BPPs than meets the eye. For if you start looking at the literature, then you can find, for example, that this peptide, the BJ-BPP10C, it protects cells, those are neuroblastoma cells, against oxidative stress produced by hydrogen peroxide. So they show that after stimulation of the cells with hydrogen peroxide, so this is the cell viability, and this is the control, this is after the administration of hydrogen peroxide, and this is after the administration of 10C alone, the peptide, and then 10C plus hydrogen peroxide, and then you have a higher viability of cells. So it seems that BPPs are not only capable to modulate the pressure, modulate the availability of bradykinin, but they have other relations or other effects that might, or other targets that might be worth exploring. So this has renewed the interest in these molecules, at least for myself. And so we might ask, what are the other putative causal relations that BPPs hold? There are other biological activities that depend on different parts of these molecules. So this is what led my first interest in these molecules. And then we published this work. It was developed by this student, Samuel Ricosta, where Bothrops moojeni, actually, when it was collected at the Ilha das Batatas in Piauí state, there were a few doubts about the identification of this snake. They were not quite sure if it was actually Bothrops moojeni, because it seemed to be a subspecies, so they wanted to analyze by LCMS to see what peptides we could find. So there were some doubts, and then we wanted to analyze the venom to see if it was actually a subspecies. We performed LC-MS studies for the characterization of peptides in the snake venom with focus on BPPs. We performed some molecular docking and dynamics to evaluate the putative binding to arginine succinate synthase. We performed the chemical synthesis of BPPs, and we tested the vasodilation using isolated red aorta and promoted by these peptides. So, as I told you, I consider this as a first movement towards this line of research. The main goal is to explore novel structure-activity relationships in BPPs. So this is a map just showing where it was collected. So this is Ilha das Batatas, where the snake was collected. This is the snake, the specimen itself, from which the venom was obtained. And I've simplified the results. As I told you, I'm a mass spectrometrist by formation, so I'm going to focus more in the structural characters of these molecules. So this is the total ion chromatogram. So as you can see, you have many peaks that were obtained from the animal venom. And this is just from the low molecular weight fraction, because we did some pre-fractionation before LCMS studies. And at first, what is interesting is that this peptide, for example, contains many putative retention peaks. As you can see, it is not, it does not elute as a single peak, and although some other peptides were eluted. So peptides presented multiple retention times, and some peptides without proline presented single retention times. So after fragmentation of these molecules, we acquired this data using information data acquisition method. You can see that the MS-MS spectrum from this specific molecule is very puzzling to analyze, because mostly Y and B ions, which are very common in the analysis of mass spectra, are not dominant. You usually have internal fragmentations, which are refragmentations, producing very intense ions. This complicates quite a lot the interpretation of spectra of BPPs. So this makes them challenging for the mass spectrometrists to analyze. So I've just highlighted two of the fragments that were being and just analyzed. So this one and this one, these are two internal fragments. So these are acylium ions that were being produced by two fragmentations of this molecule. And why do these peptides have then multiple retention times? Apparently, this has been described before, mostly by performing ion mobility mass spectrometry experiments, but then what we can find is that this is associated with the presence of proline, mainly because proline has a low barrier of rotation around the peptide bond, which produces cis and trans conformers, and that might lead to a lot or to various conformations for these molecules, which might extend even more the putative biological activities that these molecules might have, because they have many putative conformations that might be achieved. So when you start thinking about, so usually peptides are in the trans conformer and they are fixed in the trans conformer, but then proline has a low barrier of rotation, and then you might have cis conformers being formed. So we attributed to that many elution peaks to the presence of prolines, and that is consistent with the data because peptides without proline did not present that. So after that, we started sequencing the molecules. So we performed manual sequencing of the molecules. Those are the peptides that we obtain from these sample. Usually, you can find a few of the full-length peptides and a few fragments that are being produced. So what is interesting is that the most abundant peptide, which is this one, 558.81.45, in our sequencing, it diverged from the literature. It was different from what has been reported. The primary structure for this molecule contained a different internal sequence, proline, proline, glycine, while in our sequence it was proline, glycine, proline. So apparently it was either us or the literature was wrong in terms of the sequencing of this molecule. What we did was to synthesize the peptide that was reported in the literature and compare the fragmentation spectrum of the synthetic peptide that we synthesized ourselves, the fragmentation spectrum of that ion obtained from the venom, and the fragmentation spectrum of the peptide that was previously reported in the literature. And then we looked for an internal fragment that could only be generated in our sequence and not in the sequence that was previously reported. So the fragment was PK, so proline lysine, so that's an internal fragment, and then you can see here in 226, PK 226, that's the sample that was obtained from Bothrops moojeni, that's the synthetic peptide that we synthesized and characterized, and that's the peptide that was described in the literature. So the peak in 226 was absent, which means, at least in my understanding, that the structure was indeed wrong in the literature. And I don't blame them because it's quite hard to get to the sequences. Actually, what we also find by mass spec that I consider puzzling is that we found some molecules with unoxidized tryptophan. So when we performed the MS studies, so this is a MALDI data. So this is the precursor ion, 1400. So when we analyzed this peptide, we could see that it was plus 16 Daltons in relation to other previously characterized molecules, and then we went after where that oxygen was. So we started analyzing the imonium ions. Imonium ions are produced by the fragmentation of amino acids from the main chain of the peptide. And then we could find that instead of the very characteristic imonium ion of 159 that is found in tryptophan, we could find 175, which meant that 159 plus 16 is 175. And then oxindolalanine was our proposal. So this is the molecule that we found in this structure. And this is novel data, but we are unsure up until now if those oxidations they have a biological meaning or if they are artifacts. So we cannot know for sure. What I can say for sure is that when we oxidize some molecules that contain tryptophans, we don't obtain this. So we don't obtain oxindolalanine. We obtain other forms of oxidized tryptophan with a different mass. So I'm not quite sure if that has a biological meaning. But then again, it's a finding. We found oxidized tryptophan not for just one molecule, but for a few molecules. Also, we characterized several other peptides. So this is all unpublished sequences obtained from Bothrops moojeni, but then again, since the mass fits, it's all in there. It seems I'm quite confident in my sequencing, but then again, I have I need to confirm this. So I need cDNA sequencing or other source of information in order to confirm, because this is quite a lot of other primary structures of BPPs from Bothrops moojeni, and we haven't published that because we are not quite sure if that is, we need to confirm that before publishing. So this is more for the students. So if you are interested in sequencing peptides, this is almost propaganda. So this is the web page of our lab, and then we have recorded lessons of peptide sequencing that you can find under this website. And then what we can offer, then you can find lessons in peptide sequencing. So this is more to the students that are watching and are interested in finding more about peptide sequencing by mass spectrometry. Then again, we started synthesizing this molecule, and peptide synthesis, we do it in our lab. So peptide synthesis is quite straightforward, unless there is a problem, and there was a problem. Because when we started first synthesizing these molecules, the C-terminal ends in proline-proline. So we used one resins preloaded with proline. And then when we added a second proline to the resin, something happened. And then by the end, when we cleaved off the peptide from the resin, instead of obtaining this primary structure, we obtained this without the proline-proline. So that was quite puzzling, because the proline-proline was gone. It was not there to be found. And then what we found out is that this diet, proline-proline, is quite difficult to synthesize based on this method, because there is a cyclization reaction that happens when you deprotect. So this is for all those who have synthesized peptides before. There are a few here in the audience maybe. So when you deprotect the second proline, it attacks the carbonyl that is attached to the resin, and then by attacking, it creates a cyclic molecule called diketopiperazine that is soluble in the solvent, and then it just washes off, and then it releases. It leaves the resin with the hydroxyl moiety, and then the following amino acid residue is coupled. So what happens is that proline-proline just cleaves off itself. And it took us a while to find this out, but then we found out. And then we use chlorotrityl resin, which imposes steric hindrance for the cyclization. So it's quite voluminous, so it is very bulky. So instead of cyclicizing, it just doesn't cyclicize. So we synthesized these 3 BPPs. So we chose 3 BPPs from that list, synthesized and purified by HPLC for testing. So what we did is, at first the idea is to go for assays, but then we did some docking and some molecular modeling to try to find where these molecules might interact with the human argininosuccinate synthase. What we did is, after docking, we found out that there's a specific region. So we used an algorithm called dog site scorer that finds putative binding sites for molecules, and then we found out that there's a cavity just above the citrulline binding site where all peptides were able to interact and presented negative scores, which means that they were capable of binding. So we added BJ-BPP10C to test if the docking was actually identifying a positive docking for these molecules. And then we performed some simulations, and the simulations indicated that peptide binding to argininosuccinate synthase increases the accessibility of the L-citrulline binding site. But only due to the cis-trans isomerization of prolines, I think that the range of accessible conformations is much wider, and then we did just the simulations with trans prolines and not with the cis prolines. And I like to study that further in the future. So we performed some quantification of the binding affinities. This is BJ-BPP10C. So it presented a negative change in the negative free energy of binding, -9, while BPP13A, one of the synthetic molecules, presented a negative value of -8.11. And BPP-BM1, the peptide that we described ourselves, presented a negative delta G of -10. So these two that are highlighted are experimentally confirmed as activators of argininosuccinate synthase. So the BPP 10C presented 85% maximum activation of argininosuccinate synthase, while BPP 13A presented 41% maximum activation. At last, we perform some assays with the relaxation of red aortic rings, just to verify if the peptides are actually capable of relaxing red aortic rings. So this is the result for BM01, this is the result for a peptide called BAX12, and this is BPP13A. So what we found is that the relaxation of red aortic rings, BPP-BM01 was the most effective with 37% maximum relaxation at the highest evaluated dose. And it is hard to establish a direct relation between activities and putative binding to argininosuccinate synthase. These peptides have multiple targets besides this molecule, ASS, and we find that BPP-BM1 is promising. We will advance in more assays and look for experimental proof of the activation of argininosuccinate synthase. Also, we are going to synthesize oxidized versions of this molecule and try to define if oxidation is actually relevant to the relaxation. And the idea is to explore novel applications of BPPs. They might be antioxidant, neuroprotective, and what else? So there might be other activities associated with these molecules. So I'd like to thank the LASAB team. So these are all students, both MSc and PhD students, and all the collaborating labs in this line of research, and also the financing agencies, CNPq, CAPES, and FAPDF for financing us. So thank you very much.
Thank you, Guilherme, for your nice presentation. Questions?
I am a biologist as well, and I'm very curious about many things. Congratulations for your talk, very interesting.
Thank you.
Yeah, I'm guessing about too many prolines, one after the other. It's very strange because it causes a steric
hindrance.
And then how do you explain this steric problem and the flexibility to adapt to many different binding sites?
I cannot explain much. So at first, your comment is very good, actually. If you have like many prolines that are just one after the other, the change, let me try to be accurate. The isomerization of a proline to the cis conformation might hinder the isomerization of the following one. What we did find out, I just picked one result. So what we did find out at the number of peaks was somehow, I'm not quite sure if it is, if we understand well enough, but it is somehow related to the number of prolines that the molecule has. So the lesser the prolines, less peaks were observed. But I think that chromatography is not the best technique for that. So what we aim to do is get the synthetic molecules and then use ion mobility to try to characterize, based on the collisional cross-section, these molecules, to try to evaluate how many conformations that do these molecules have. So what I find puzzling is that these molecules are usually multidimensional. They have many interactions with many receptors, and I think that we settled too soon for the idea that BPPs interact with ACE. So they inhibit ACE. And I think that we settled too soon in this idea, because if only ACE interaction is relevant, then why sequence conservation? It doesn't make much sense to me. At least to me, it doesn't. So there might be some other targets, and they might be related to this high conformational flexibility that these molecules have. But I have too many questions and too few answers at the moment.
Have you compared, I don't know, you didn't do the docking to the ACE, the BPPs to the ACE?
We did not. We went straight to ASS.
Okay. But if you compare the structure of ACE and the ASS, argininosuccinase, do you see any similarity or analogy in the structures they have?
Curiously, it seems that the proline-proline moiety at the carboxy terminals is also relevant to the interaction with argininosuccinate synthase. So the same moiety that defines the interaction with ACE, apparently, and it was conserved in all the dockings and the dynamics that were performed, the carboxy terminals of the peptides were in the same position in the argininosuccinate synthase. And it has been described, because it has been done by some colleagues from Butantan. They did some modifications in this molecule, and they have described that actually the proline-proline moiety is indeed important. But then again, some BPPs interact with ASS and seem to activate ASS, reduce the Km for the substrates, and some apparently don't. And that doesn't mean directly that these molecules will work as vasodilators or not. So it seems that ASS is once again just one target, another target besides ACE. So there might be others. Apparently, yeah. Many causal relations.
Congratulations, Guilherme. I ask you if did you try the ring of aorta without the endothelium?
No, we didn't. At this moment, no. But
Yes.
we intend, now that we have synthesized this molecule and we have quite a lot of them. This is I have a peptide synthesis lab, so we have synthesized quite a lot of this peptide, and we are sending to our collaborator to do these assays. So he's going to remove the endothelium and perform these assays.
It can give you some more information
For
about
sure.
the site.
Yeah.
Thank you.
Thank you, Professor.
Any other question? So, thank you very much, Guilherme.
Thank you. Thank you.
I will call our next speaker, Edhan